Okay, so while you're enjoying your pizza, let me present the speaker. If you're like me, when I was in grad school, learning about biological structure meant protein crystallography, which is essentially what you get when all the proteins very nicely line up in a row and agree to be interrogated by an X-ray beam. And of course, biology isn't really like that, as we know. And what Peter's going to tell us about today is sort of one of many techniques that you can use to study proteins when they want to do things like rotating and flopping around and interacting with other molecules. So Peter got his PhD at the EMBL in Hamburg with Victor Lamson, and was then a postdoc with Spies-Dowder at both Brookhaven and Argonne, as I understand it. And I got to know him when he came here to LBL to work with Paul Adams on the Phoenix program for protein structure refinement. He was with us for several years, and then he moved over to the Berkeley Center for Structural Biology, where many in the world have gotten to know him as a beamline scientist there. But he's continued his interest in biological structure. And just to give you a flavor for the things Peter thinks about, we sat down with Cheryl Kerfeld a couple of years ago and asked, how can we use the new light sources like the free electron lasers to study carboxysome structure? So these are small organelles which do carbon fixation in certain bacteria. Well, do they crystallize? No, it turns out they don't. Many people have tried. Well, can we line them up in a nice row and put them in the X-FEL beam and get single particle diffraction? Well, that turns out to be very difficult too. So Peter says, no problem. We'll just study them in a disordered state in solution. And that's one of the things we'll be hearing about today. So Peter.
Thank you very much, Nick, for the kind introductory words, and thank the organizers from PBD to give me the opportunity to present this work, and thank everybody here for showing up for the pizza and coping with me in the meantime. So I'm talking today about fluctuation scattering, and it's basically the femtosecond extension of small angle scattering, and it's a technique that most people are familiar with. And I'm a research scientist in PBD, and I do stuff in the Berkeley Center for Structural Biology. So solution scattering, as Nick alluded to, can really provide you with very important clues on how proteins behave in solution. Moreover, solution scattering, small angle scattering, wide angle scattering, it can answer questions that other techniques cannot really answer or are very difficult to answer with other questions. For instance, there is a great paper where people from just across the bay in San Francisco work on HSP 90. They have shown that using solution scattering methods, they have shown two distinct conformations in solution, and they derive really important biological function from this. Same thing with work done by people from John Tainer's group and collaborators on this structure here, and there's a paper there. Now, solution scattering is a high throughput technique. You can really automate things nicely, and people have done great things with solution scattering. There is a bit of a challenge though, and the challenge with small angle scattering is that the amount of data that you have the amount of data that you get from the experiments has a very inherently low information content. And using Shannon's theorem, people say that typically you get, well, maybe 9 to 15 individual independent parameters from a small angle scattering curve. And that's of course not very nice. If you look at a system like lysozyme, it has 1,001 atoms roughly. And if you want to describe that and the dynamics of that, and you only have 9 independent parameters, that is considered to be a bit of a challenge. Still, people have done amazing things with this, so the situation is not lost. But I think everybody agrees that the more data you get, the better it is. Now, it turns out the reason why the information content in the data is so low has to do with the fact that while you're taking your snapshots, your molecules stumble around in solution. And what you get basically is a physical 3D averaging of your scattering patterns, and that washes out any details, and the only thing that you end up with is a uniform, angularly uniform scattering pattern, as you can see here. What happens when you don't have a lot of data? Well, it becomes really easy to overinterpret your data. Moreover, sometimes structural ambiguities cannot be really resolved from the data alone. And this is a great example. This is a data set provided to me by Rob from his BioISIS website, where the data itself here in black, if you try to do Ab-initio structure solution, you get either looking something like a crooked banana, something that looks like breakfast, or not sure what that is. But these structures are more or less equivalent. Only when you start to apply symmetry, external information, you'll be able to get to the right answer. And that's of course a problem, because when you want to use shape to assist you in interpreting biology, you don't really want any ambiguity, so you need to reduce them as much as possible. So, in order to get more data so that we can understand biology better, we want to have more independent data points, and in principle we don't want to have them, we want to of course have them at higher resolution, but also in each resolution bin we want to have more data points. Smallangle scattering is basically you have your 3D structure, your row of R, and that turns into a 1D data set, just a mean intensity is a function of resolution. Whereas in crystallography, you have all the data that you need, you go to a 3D data set. Now, the more data you have, the less ambiguities you have, and the better the models will be in the end. Now, I spoke earlier about, that was here, that the reason why the information content is low is because of orientational averaging during the exposures. Now, if you want to get more information in your scattering patterns, what you need to do is you need to make sure that the scattering pattern is taken while the molecules don't move. Now, there are two ways of doing that. That is, A, you can immobilize the sample, or B, you can make shorter snapshots. Taking shorter snapshots is really the way forward. And I can go over the numbers later if you want. But taking shorter snapshots and not freezing your samples is I think, a better route. This is what you get when you have a limited number of particles in an X-ray beam and you shoot at it with a 80 femtosecond pulse. You see these nice fringes here, and this is because of the fact that you have 2, maybe 3 particles in the beam in the beam, and these fringes occur because of that. Now, shooting below the rotational diffusion times is hard. You need to have a very short pulse, and the way to do that is to use a free electron laser to generate enough photons in that short amount of time. The various options there. The one that is most convenient at the moment is the NGLS in Stanford. Something that would be more convenient, and there's actually a very good reason why we need the NGLS for this kind of work, but I'll come to that later. Is to use the NGLS, which is a light source that we hope is going to be built here on site in the future. Now, when you shoot images at below rotational diffusion times, you get Speckle patterns, and you see an example of this here. This here is an image of a biological sample in solution. This is only a small part of the detector. The detector is much larger than this. This is at low resolution, but you can clearly see visually that you have Speckle. You have tiny intensity fluctuation in the image, and it's non-angularly uniform. So this is different from Smallangle scattering data. You have a whole lot of nonsense here. I can talk about that later. But the main thing here is that you have Speckle here, and that is because you have multiple particles in the beam, and they don't move around, they don't tumble around, and that's why things are not angularly uniform anymore. Ideally, you would like to do this on a single particle. If you have a single particle, you just have to figure out its orientation, and then you can pull off some tricks and maybe get the structure. Turns out that getting a single particle in the beam is not very easy. and having multiple particles in the beam is much easier to do. So when you have multiple particles in the beam, what Nick, basically this goes back to what Nick said before with the carboxysome discussions with Cheryl. When you have multiple particles in the beam, still the situation is not lost. What you can do is you can, rather than looking only at the average image, you can look at the angular autocorrelation. And that's basically a cheap trick to overcome the fact that you cannot untangle these patterns to get the scattering patterns from the single molecules. If you have 20 molecules here, all with unknown structure, unknown orientation, unknown position, it's going to be really, really tough to separate every single bit out. It's just not going to work, so you need to do something else. And the something else is the angular autocorrelation. I'll try to explain you how that works. If you have a system that looks like this, if you have a circle that looks like this with the two spikes on top, and you want to compute the angular autocorrelation of this, you basically take this one and a copy of itself, and you rotate it around, and then you basically you look if you multiply the two patterns whether or not you get out something that is zero. Okay, so in this case, the only time that you get significant overlaps is if you rotate this pattern just by a wee bit to the left and a wee bit to the right, and you see that here. And then if you average these, or if you compute the final autocorrelation looks like this. And this is really nice. Now this is for one particle. Now it turns out that if you have more than one particle, you start getting things like this. So you start off with this, the autocorrelation with itself, the angular autocorrelation where you don't separate things is here. Then you start moving things nicely you rotate things slightly to the right, and you get starts intersecting here and there, you get this spike, and you rotate it further, etc., etc., etc. And then in the end, your autocorrelation looks like this. Now, if you compare this to the previous autocorrelation that you have here, this is how it looks for a single particle, and this is how it looks for the two-particle system. We have an extra feature. We have this extra feature here, right? We have these two ears here. Now, these two ears here, they come from this opening angle here, and that is of course nasty that you have that, but not all is lost, because we actually do these experiments many times, and every time we have a completely random configuration of molecules. So you have one time it looks like this, the other time it looks like that, the other time it looks like that, the other time it looks like that. And what happens is that this spike here will be randomly oriented here on the circle, and effectively what you get is on average you get a thick baseline with the single particle autocorrelation on top of that. And that's how you get your angular autocorrelations. Now, the mean angular autocorrelation that you get from the experiment by basically doing a lot of snapshots, and for each snapshot you compute your angular autocorrelation, you can relate that angular autocorrelation to the structure, you can relate the structure to the angular autocorrelation in a relatively simple way, and so we have methods to compute that. You can see here some model studies. This is something we published in ACTA a while ago. We use 3D Zernike polynomials. I won't go into that because people will run away. But we can take models, we can expand them at different resolutions, and still get fairly good performance of computing how the data should look. Now, how do we do the experiment? The experiment is relatively simple. So first you need a free electron laser, and second, which is depicted here by the blue-purple line. And then you have particles that you inject. They hit the X-rays, and you basically get a speckle pattern on a detector. And this data is what you this is the data that you analyze. Now, the FEL and the jet intersection is right here. So this is here the nozzle with the liquid jet being spat out in vacuum, and this is here basically a liquid column of dilute protein solution. Now the X-rays hit the solution, scattering patterns are recorded, and then subsequently you reduce the data and you solve the structure. That's how it works in theory. So sample delivery is not easy. I don't know anything about that. These are the collaborators from Max Planck and SLAC as well as ASU that solve this problem. Recording scattering patterns. Well, detectors are cheap. Well, not really, but we have detectors. We have detectors that can do this, but detectors also have their flaws, and that makes data reduction difficult. And I'll show you some problems that we have. So the data processing is difficult because, well, we live in the real world. First, we wrote simulations, and simulations are really clean, and we can deal with simulated data really nicely. And then as soon as you do an experiment, the whole world collapses. Turns out that we actually see scattering actually from the jet on the image as well, and I see here. Well, it's difficult to say. I think this is the best. This is one of the better images where you can see it really clearly. You have this jet scatter. There's an enormous amount of jet scatter in between the detectors. You don't see that because the detector has two halves that are physically separated. So the bulk of the jet scatter is not even on this image here. But there's another component of the jet scatter that is lying here. And because we compute angular autocorrelations, because we try to correlate each pixel with every other pixel in the detector and then compute averages, we only want signal from protein. We only want contributions from protein. And when you have jet scatter, that's really strong and dominates the signal that you get. So we need to get rid of that. There are two ways of doing it. One way is to redo the experiment and see if you can get anything better. But since we only get 5 days a year or even less, that's not really feasible. So the second option is sit down with the postdocs and figure out whether or not we can solve this. And I think we are doing a really relatively good job at the moment. So we can look at the images and decide where the streaks are. You might say, okay, maybe this is something you need to do by hand. And yes, if we have, well, enough people, we can do it by hand. But enough means 80,000 people, something like that, because we have over 100,000 images. Yes, Bob. So if you scatter off of the droplets, you have to make sure that the X-ray beam is smaller than the droplets because you don't want to have any signal from the droplets. The other thing is that we need a lot of images, so we really need a high hit rate. And if we scatter off of the continuous bit of the jet, we're always hitting the beam. So what you see here, this is done like that, and then the biggest streaks are in here, and the small streaks are there, and we seem to be able to correct for this quite, in this particular case, quite well actually. For an experiment that didn't go that well, and it's difficult to see exactly how far this extends, is that in this particular case we have much stronger jet scatter here. And the problem is that the jet scatter is not always at the same place, and it varies from shot to shot. So that makes it really difficult to apply a global mask. If we were to apply a global mask, we would lose 75% of the data, period. So we need to do shot-by-shot masking, and we need to do that as conservative as possible because we don't want to lose data, because we need a lot of images in order to get rid of noise. So this has some automation challenges that need to be solved, but I think we're making really good headway with that. Now, once you have eliminated all your artifacts, you can compute the angular autocorrelations, and that looks something like this here. This is real experimental data from virus data. From viruses, I'll talk about that a bit later. But once you have these curves, just for my pleasure and your torture, we need to expose these. We need to decompose these patterns into a polynomial series with Legendre polynomials. And there are good reasons to do that, but just accept that this is the right thing to do. But we get these expansion coefficients. And it's basically you get a whole stack of curves like this, and you put them in the magic box, and what you get out is a set of curves like that, and you can go back and forth between these two representations, and you don't lose anything. So this is basically what the experiment gives us. In black, this is B0, what we call B0. B0 is equivalent to small angle scattering data. So small angle scattering data is a subset of the data that you get using this technique. Turns out that you don't have to only correlate pixels that are in the same resolution shell. You can also correlate pixels that are in different resolution shells. And basically what you get, so here this is roughly a 2D data set, right? SAXS is one-dimensional, but we have more curves, so we add another dimension to it. It turns out that if you start computing correlations between other pixels, you basically have a whole stack of all these surfaces here. And what you get is you get resolution and expansion order dependent expansion coefficient surfaces. So you get a whole lot more experimental data when you do your small angle scattering below rotational diffusion times. And that's good, because we really need to have much more experimental data in order to be really confident about what's happening in solution. So summary, what we do is we have proteins in solution, we shoot it with a free electron laser, we get lots of data, we remove the artifacts, calculate angular autocorrelations, get the coefficients out, and then out pops the structure. More or less, not really. Let me tell you how we can do that. So, small angle scattering, people have been doing this for ages. They have basically using dummy atom approaches to basically construct a real space model that fits with the SAXS data, and we can do more or less the same thing. And it's a Monte Carlo based procedure where we basically start off with an arbitrary shape in real space and then modify it in any way that we like, as long as it improves the fit to the data. These are reconstructions that we did using synthetic data, and the associated models are there. This is not really interesting, but please keep this model in mind here. I showed you this before. So first of all, it's important to state that during the Monte Carlo optimization that we have done in order to recover the structure from the data, we did not use any explicit real space restraints or symmetry constraints in the reconstruction. So we don't explicitly penalize connectivity or say non-connectivity. But we do obtain connected structures. Turns out that if you look at the map correlation in real space to the known object and to what we get, and this is for the donut protein, peroxyredoxin, this breakfast fellow here. If you use the fluctuation scattering data set, synthetic data, you always end up with a donut. If you use small angle scattering data, you end up with the crooked banana, which is really interesting. So basically, by having more information in your data, you eliminate, at least in this case, you eliminate ambiguities. And I've got an animation here that shows you the progression of structure solution. And you start off with a sphere, and then it just crashes. It goes to the. Let's see here. We do that again or not? Let's see if this works. Yeah, well, and it doesn't like me today, I think. I move. Yeah, okay, you can see here how this is how it looks overlapped with the structure. This is just a single run. You need to do this many runs and see if you can improve on the result, of course. But again, we did not use any symmetry or connectivity restraints in the process. So the whole reason why we want to do the experiment like this is to get more information. So as I've shown you, ambiguities are reduced, which is really important. And when model data is available, this information gain or the ambiguity removal is even more striking. And what you see here is a RMSD versus a mean chi-square per Shannon pixel, so to say. It's a normalized chi-square, so that we can put the scoring function for fluctuation scattering and small angle scattering at the same scale, so that we don't artificially inflate this curve because we have more data here. So it's basically the mean chi-square. And what you see here is that this scoring function here, the chi-square for fluctuation scattering, goes down steeper, closer to 0 angstrom RMSD as it does for small angle scattering alone, which is an indication that you have more information in the data. And I can do the same thing here. This is simulated data, and there's supposed to be a bit of a wobble of a normal mode here. It's unfortunate that it's not coming out, but these are two normal mode axes, normal mode 7 and normal mode 8, with a perturbation of -0.5 to 0.3. And what you see is that the energy landscape, or the scoring, the chi-square landscape, or something that looks like a chi-square landscape, for fluctuation scattering really goes down very steep, whereas for small angle scattering it is relatively flat. Again, the small angle scattering curve here is just for the small angle scattering data, and this here has up to expansion coefficients, I think 12, so it has much more data. So if I divide that up to put it on a similar scale, and do a smooth approximation around the midpoint, you can really see that there is much more information on average in the data in the individual data points than if you only have SAXS data, and that's the real reason why we want to do these things. So in the end, the average data point contains more information, and that results in a better discrimination of likely versus unlikely structures, and that's really important. For instance, if you want to look at membrane proteins and see look at structural changes, having more data, apart from only the SAXS data, actually having all the higher order curves as well, might actually be very useful in determining exactly what's happening. Okay, this is all theory and some practice. Can we actually do this experiment? Because it's not at all trivial. I told you we have done some experiments. We have done some experiments on large particles, because the larger the particle is, the easier it is at the moment. We have done this at the AMO instrument at the LCLS in June or July last year. I think it was July last year. This is an example of a virus in solution at 520 electron volts. This here is what was supposed to be a carboxysome, but I think it's a virus. And this was aerosolized data. You've seen this here. This is how it looks. Now we've done this on CRO-V. It's a large virus, somewhat like the Mimi virus things. And the analysis on the CRO-V virus data really indicates that this method it's not all just theoretical bogus. It's really possible to do this. So we see here examples of angular autocorrelations at a fixed resolution shell. And this here is the autocorrelation that you get when you only use images that have a single particle. This is when you have two particles. This is three particles. Three particles. This is more than 3, and this is what you have all. And in all these cases, all the patterns look the same. This is exactly what theory says it should be, and this is what we get. So it's not a surprise, but it's really encouraging. This is the experimental data that we get after we reduce it. It looks really good. There is something really funky going on right here. I think we know why that's the case, but we just haven't fixed it. I'm not sure if we know why, but we need to do something about it. So, of course, I showed you the algorithm that we have. So we did let this loose on a subset of the data. We used B0 and B2, so it's SAXS plus a little bit of extra stuff. And what we get out is something that matches the curves reasonably well, has the right size, and has a hole in the middle. When we show this to our collaborators, I said the model is wrong, there shouldn't be any hole inside. Well, we have to see. I have data. She has an opinion. She might be right. So this is streamed online. I'm sorry, Ilma. Sorry. So we also did stuff with carboxysomes. We have single particle aerosol data, and after cleaning it up and doing size selection, we have an amazing 27 images. This is not minus 27 images, but it doesn't take that much from where we are now to get to minus 27 images. Also, we have multiple particle solution scattering, and we really have images from the Horn of Plenty there, and we have loads and loads of these patterns, and a lot of them, there's a sizable number of patterns that actually contain features that really mess up the analysis, so we have to be very careful in what we include and what we don't include. If we look at the autocorrelations from the single particle images, it looks relatively good. We get the autocorrelations that look like this. It's fairly noisy, but it's not impossibly noisy, and I'm actually really happy with this. If you do the BL expansion, this is what you get here. The SAXS curve actually matches really nicely what we get from synchrotron data. And in green, this is B2. We have this double hump here, and we have this double hump right there. And we have seen this double hump feature in some model systems as well. So I'm really happy with this. The liquid jet data, and that's about 20 particles per shot roughly, is cleaning up slowly. We have to. It's not easy to process this. We basically we are testing various algorithms, and because we need a lot of images in order to get convergence, it's not easy to go through the processing cycle of methods development, because if testing the algorithm takes a day, then yeah, you don't have a turnaround time of methods within half an hour, so it's a bit more difficult. But then again, the angular autocorrelation here looks pretty good. Well, you could say it doesn't look very good, Peter, but actually I think it's really promising. First of all, we have a peak in the middle here. I can tell you that we haven't seen this peak in the middle when we were doing things wrong. Also, there's a signature of a hump right here, and there's a signature of a hump right there, and which is nice because we expect there to be a symmetry axis right around here. This here is, I think, signature from the streaks, and we just have to find a way to get rid of that, and that will take some time. But we can take the data as we have it, and we can fit things to B0, B2, and B4 all at the same time, and then we can get out a model. And it looks highly detailed, and we are most likely overfitting, and even the fitting is not really fantastic yet, but overfitting is the way forward, because later on you average things and then things look nice, and this is the way things are done in Smallangle scattering, and I don't see any reason why we need to do something else at the moment. So this is a work in progress. Now, why is this relevant to the NGLS? This was all funded by an LDRD for development at NGLS. Now, we've done this at the LCLS, and it works really nicely. Now, if you go to smaller systems, like 150 kilodaltons, you need about 100,000 images in order to get to inverse angstrom of 0.1. And that's maybe 15 minutes at 120 hertz. Now, because scattering power falls off with q to the power of 4, if you go to higher q values, you really need a lot more images in order to beat counting statistics and well, basically scattering from solvents. And that really is very difficult. So you need much more images, tens of millions. That's really difficult to do at the NGLS because beam time is really costly. It's also really difficult to do at the NGLS because it's not built yet. But if it is there with a high rep rate, like in the kilohertz, you can do this, you can get tens of millions shots really easily, and it really doesn't take a lot of time, and it shouldn't take a lot of sample. Now, by doing these experiments at the NGLS in the water window, 3 things happened. First of all, you get a very significant contrast enhancement, and you can go to the X-ray data booklet, and you can look at what the difference in contrast is if you are at hard X-rays like 10 keV, and if we set that if we define the contrast gain to be 1 here, if you go to the water window, you have a contrast gain of a factor, a squared contrast gain of a factor of 4, yeah, which means that your signal is going to be better, which is really great. So doing this in the water window is great, and you don't have to sacrifice resolution because Smallangle scattering is not really about resolution anyway. You can easily get there, and the NGLS will be able to do that, and you at nice contrast. You can do this at really killer rep rates. You can get as many images as you can burn postdocs, so to say. And more importantly, we don't have to wait for the hard X-ray free electron laser to be constructed. You can do this basically from the first day that light is obtained, which I think is really important because as we all know, funding is always uncertain, and if we have a machine that is there that can do all these things, we really need to be finding good uses for it in biology, and I think this is a killer application. Now, this is more or less what we've done. There's things that we're working on. Just to preempt questions, people say, well, don't you want to have single particle stuff? Yes, you do want to have single particle stuff. Actually, you can bootstrap single particle structure determination in a real easy way. You take all the data, all your single particle data, you get your fluctuation scattering data, your autocorrelations, you get a low resolution structure solution, and then you can actually get a model. You can actually get an estimate of your 3D diffraction pattern from your model, and then you can go back and you can interrogate your data, and you can do image registration. You can figure out what orientation is this, and this is really nice. I think it's much more important that you can actually go back to your data and can say, is this garbage or not? Because what we've seen right now is that there's so much garbage in the data that actually being able to interrogate images that you have and figure out whether or not things are outliers could be really important in these procedures. It turns out that we understand data from mixtures as well. Mixtures from fluctuation scattering behave in exactly the same way that mixtures from mixtures in small angle scattering behave, which is nice. That means that we can untangle the data under favorable circumstances for a mixture of conformations, and that basically means that we can do time resolved work if we want to. Now, when we have more data than we have right now, and we can already do this with small angle scattering data, but we can also start moving into structural refinement techniques such as normal mode refinement. If you have more data in each resolution bin, you can actually start doing things like cross-validation to make sure that you do not overfit. This is more or less the end. I'd like to say that free electron lasers provide novel opportunities in solution scattering. You can get information-dense data that will allow you to improve your structural models. Development is ongoing, and many challenges still need to be resolved, but I think we're well on our way to figure out where we need to go, and we have good data to show that we will be able to get where we want to go. We have done proof of principle experiments on viruses and carboxysomes, and that shows that this is indeed feasible. But it's going to take some time before this technique is going to be part of your tool chest in order to solve your problems in biology in a routine fashion. This is work that many people have contributed to. I'd first like to thank Billy and Eric, postdocs currently, and I'd like to thank Hai Guang, who's a postdoc now with John Spence, who have been really playing pivotal roles in this work. Cheryl is the person who has been instrumental in pulling everybody together to get these experiments going at the NGLS. Ilma has played a major part in it as well as where is she? Petra Frommer. I also like to thank Abbas Urmas and Dilano Saldin for help with data analysis and my theoretical understanding. Money comes from LBNL, LDRD, and recently Cheryl obtained NSF funds to work on this as well. And I'd like to leave it at that. Thank you.
Do we have any questions from the audience?
So you did a carboxylic data on single particle and multiple particles. Is that right?
Yeah.
How did you adjust for single particle versus multiple particle in terms of classification of the sample to
So it's two different experiments. So the single particle one was aerosol data, was an aerosol. And the multiple particle was a liquid jet. So it's two physically different sample delivery systems. The aerosol it's really a very wide cone of particles that is being delivered, and you just have to be lucky that the X-ray beam hits your sample. Whereas the liquid jet is a very, very thin jet that you can place very accurately. Of course, the single particle data looks very nice because you don't have any solvent background. Yeah, this is basically a particle in vacuum. We hope. As of course, there might be crud on the outside because the water evaporates, but the contrast is really nice. But the problem is that the hit rate is very low. Here you have less contrast. You got streaks, but you've got much more data to deal with. And I think this is the way to go for the fluctuation type of experiment. This data here, we have single particle data from viruses as well, and that was done in the liquid jet. We didn't spend a lot of time on aerosolized viruses.
My question concerns the anomaly that you mentioned at low values of Q. I would expect for the kinds of particles that you're looking at that there are flexible appendages that are sticking out into the liquid phase that will be more disordered than parts that are internal to the particles. Could that be an explanation for what you're seeing there?
In this case, I don't think so. I don't think that this is the problem. This particular problem here most likely has to do with the way that we fit the data. The problem that you're talking about with flexibility. So actually, this virus, I think there are no hairs on this virus. So it's basically stripped empty. So there is on the outside nothing really majorly floppy. The nice thing is that handling floppy particles is something that's well, that's the interesting bit, right? That's how you get your biology, and this is the bread and butter of the Smallangle Scattering community to handle systems like this and to get the most information out of the data. So when you do have systems that are floppy, by getting data like I propose, you will have more data so that it's going to be easier to distinguish nonsense models from real models. People do fit multiple models to the data, but there's always the question: are you overfitting or not? And by having more data, you can be more sure that you are doing the right thing.
Other questions?
I know you're saying that it's going to take some time for this to be a general procedure, but you're looking forward to the future where you're going to recommend that everybody do this. So it sounds like at the LCLS, 10 angstrom resolution is what you expect, and at the NGLS, maybe 4 angstrom?
No, so we have done this at the moment on particles that are 100 nanometer. Yeah, we are trying to do this on particles that are 50 nanometers, and the smaller the particles get, the more difficult we think it's going to be in terms of signal to noise. And one of the biggest problems is at the moment that we don't have any good ways of doing high throughput data processing and reliable ways of getting the data out. We are working on that, but at the moment, having a result like this is not going to be sufficient. Results like this are really good and really great, but again, this is aerosol data. So we need to make various steps forward, both in the analysis but also in the sample delivery. And one of the things that we are doing this June is to use the microbeam at CXI with hard X-rays to see if the jet scatter is going to be reduced, and maybe data processing is going to be easier then. But we first need to. Well, I think we first need to be able to understand model systems before we are going to ruin months of people's work that have been working on getting the precious biological samples ready. So it will take some time. And then also, well, beam time at the LCLS is not easy to get. So that's another problem.
Is there any way this can be a parasitic experiment using somebody else's beam?
Yes. So yeah. In principle, if we know how to, if everything is squared away with sample delivery and experimental setup, there's really no reason why you should not be able to wheel in an end station and use the beam that is coming through at the other side. It should be feasible.
Peter, I'm curious about if you have a stream of particles, many particles, which are in different orientations and may actually be in different states. They may be flopping around in different conformations. Can you derive both of those? Let's say you have 2 principal conformations. Can you derive them from the same data set with your algorithm, or
not from the same data? Well, yes and no. No, the best way, if you can pester your system into change the stoichiometric behavior of the 2 component systems, you can collect data at different positions of the equilibrium, and then you can use very simple linear algebra to figure out exactly how the individual components look. This is not magic. This has been done in Smallangle scattering for ages. We can do the same thing on the data that we have here. And that is, I think, the best way to do it. If that's not possible, what you can do is you can just take 2 models and you can see if you can tinker 2 models in such a way so that it makes the data fit. But if you have 2 models, what you do is you double the number of parameters that you have. And if you just look at the chi-square value, reduction in chi-square value, that's not really fair, because overfitting becomes really easy and chi-square values go down naturally as soon as you start adding more parameters. So you have to be a bit careful. But again, people have been doing this with some success in small angle scattering, so I don't see a big problem there.
I think we have time for one more question. All right. Thank you very much for coming. I appreciate your attendance. Our next talk from PBD will be here on Thursday, the 4th of April. That's going to be a talk by the lab librarian telling you how the lab library can help with your research. Our next Science and Tech talk will be on Tuesday, April 23rd, and that will feature Param Veer de Hall talking about K-base. So hope to see you then.